Hello everyone, we made an introduction to microscopy in our previous video. We talked about units of measurement used in microscopy such as the micrometer and nanometer, and we talked about two very important concepts in microscopy: magnification and resolution. Well, magnification, as you remember, is the number of times larger an image is than the real size of the object. So magnification does not necessarily provide any clarity of the sample. On the other hand, resolution or resolving power is the ability to distinguish between two objects very close together. So, which practically means that the higher the resolution of an image, the greater the detail that can be seen. In other words, if 2 points cannot be resolved, they'll be seen as 1 point. So in practice, resolution is the amount of detail that can be seen. Well, in today's video, we'll shortly talk about two types of electron microscope, and then we'll compare and contrast the light microscope and the electron microscope with each other. There are many questions in paper 1 comparing the features of light microscope to electron microscope. So let's talk briefly about electron microscope. Well, electron microscope started being used around 1950s. Remember, the name of the microscope is given according to the type of radiation the microscope is using to view the sample. Light microscope uses visible light radiation, while the electron microscope gets its name that it uses the electron beam. As you see in this picture, electron microscope is not portable. But how does an electron microscope work? How can you use an electron beam? Where do you get the electron beam? When a metal becomes very hot, some of its electrons gain so much energy that they escape from their orbits, like a rocket escaping from the Earth's gravity. And free electrons behave like electromagnetic radiation. So they have a very short wavelength. The greater the energy, the shorter the wavelength. Electrons are a very suitable form of radiation for microscopy for two major reasons. Well, first, the wavelength is extremely short. Second, because they are negatively charged, they can be focused easily using electromagnets. So for that reason, you can see that the electromagnet has a long tube. So let's briefly outline how an electron microscope works. So no matter the shape or the design of the electron microscope, all the electron microscopes have an electron gun on the top. It's the part where the electron beams are emitted to produce a beam of electrons. And all the electron microscopes also have electromagnets. And these electromagnets, since the electrons are negatively charged, they direct the electron beam in the tube either onto the sample or through the sample. And then eventually the image on the electron microscope is reflected on a screen. So that's again one difference between a light microscope and an electron microscope. As we observe a specimen in a light microscope, the light rays directly enter into our eye. An image is formed in the eye and then in the brain. But in electron microscope, image is formed first on a screen. That's one difference. And secondly, the specimen needs to be specifically treated to be observed in electron microscope. And this treatment of the specimen requires some harsh chemicals. And these harsh chemicals kills the live sample. Another important difference, using electron microscope, it's not possible to observe live sample. And the treatment of the specimen with harsh chemicals also may form some artifacts in electron microscopy. So an artifact is any deviation from the real shape or structure of the image. Two types of electron microscope are now in common use. The transmission electron microscope, or TEM. That was the original electron microscope developed. And here in transmission electron microscope, the beam of electrons is passed through the specimen before being viewed. And only those electrons that are transmitted, that pass through the specimen, are seen. So this allows us to see thin sections of specimens and then to see the inside the cell. Well, in scanning electron microscope or in SEM, on the other hand, as the name implies, the electron beam is used to scan the surfaces of the structures, and only the reflected beam is observed. Here you can see different micrographs taken by scanning electron microscope and transmission electron microscope. So in scanning electron microscope, we get a surface image. We get a surface image that has a very high magnification and resolution, and we also get to see a three-dimensional appearance. On the other hand, in TEM electron microscope, Since the electrons pass through the specimen, we get very fine inside detail of the specimen. Okay, transmission electron microscope, very fine detail, right? Very high magnification and very high resolving power, and three-dimensional surface appearance of the paramecium taken by the scanning electron microscope. Okay, let's check out some questions. Here is a multiple choice question from June 2004. What is responsible for the high resolution of the electron microscope? Remember that the resolution depends on the type of radiation the microscope is using. So electron microscope uses electron beam. Another question. What's the resolution in nanometers of an electron microscope and of a light microscope? So the resolution depends on the type of radiation the microscope is using. So the light microscope uses the visible light, which has a wavelength between 400 to 700 nanometers roughly, and the electrons have far smaller wavelength. So the numbers may vary in different resources, but according to Cambridge and past paper questions, the resolution of an electron microscope is 0.5 nanometer and the light microscope is 200 nanometers. So these are the numbers that we need to remember. Next question. What describes the features of an electron microscope? So the magnification, the resolution, and the type of the specimen is asked in this question. So in such multiple choice question, you can use the method of elimination. Remember that due to the harsh treatment of the specimen in electron microscopy, this specimen can never be live, so we just cancel out B and D. You don't even need to bother yourself thinking about all these numbers. Remember from the previous question, the resolution in electron microscopy is 0.5 nanometer, so that should be our answer. In a light microscope, the light passes through the objective lens, and the highest power objective lens is 10 times magnification. And also light passes through the ocular lens or the eyepiece lens, which is usually 15 times. And the total magnification in a light microscope would be 1,500 times. Well, there are also some eyepiece lenses which might have 16 times magnification or 10 times magnification, but that's the number we usually commonly find in AS questions. What about the electron microscope? Electron microscope has a magnification of 200,000 times, which is this number. Well, here is another question from paper 2 of June 2010, which will also help us to make a brief summary of the features of the electron microscope. So just one disadvantage of the electron microscope compared to the light microscope for the study of the cells. But what disadvantage? I mean, electron microscope has a higher magnification, has a greater clarity, resolving power. But is there any disadvantage? Well, due to the way the electron microscopes operate, there are certainly disadvantages compared to light microscope. Well, first of all, electron microscopes are very expensive. The maintenance of electron microscope is also not easy and also expensive. You need a very special room that you can set the temperature. It requires more electrical power. The electron microscope is sensitive to external magnetic fields. And electron microscope is difficult to operate. You definitely need a technical training to use the electron microscope. A sample preparation is way more difficult compared to the light microscope. In light microscope, you just take the sample, put a drop of water, put the cover slide, and it's ready. Well, in electron microscope, specific chemicals are used. It takes longer time to prepare the specimen. Electron microscopes are not portable, so you're just confined to some specific locations. And due to the fact that the sample is first treated with harsh chemicals during the preparation and then mounted in vacuum, this may distort the delicate shape of the specimen and forming artifacts. This is also a disadvantage of the electron microscope. Let's compare light microscope and electron microscope. First, starting with the source of radiation. It's visible light in light microscope and electrons in electron microscope. Magnification. It's 1500 times and 250,000 times in electron microscope. Resolution, 200 nanometer. 0.5 nanometers in electron microscope. Light is focused in the eye, images formed on screen. Live sample can be observed, and electron microscope only dead sample. Light microscopes are cheaper, electron microscopes are expensive, light microscopes portable, electron microscopes are not portable. Well, I think we've covered the most important features to compare the light microscope and the electron microscope that we need for the AS Biology exam. That's all for this video.